Hi everyone, it's Kim Curry here. This video is going to take a deeper dive into your mRNA transcripts and how we can use the NCBI graphic view to dive a little deeper into the details of that transcript. First, I want to show you from your textbook figure 12.21. This is showing a pre-mRNA transcript at the top here, and it's showing both the introns and exons in the pre-mRNA. After alternative splicing, there are two different variants that are shown in this example. Although this rat alpha tropomyosin has many different variants, as I will show you in the NCBI database. One thing that, I guess, common misnomer about mRNA sequences and exons that are included in the mRNA sequence is that those exon sequences are included within the mature messenger RNA, but it doesn't mean or guarantee that those sequences, those exon sequences, are then going to translate into an amino acid sequence. So what we're going to do in this video is to take a little deeper dive into that mRNA sequence and ask ourselves, well, where exactly along this transcript is the translation start site? And where does translation stop? And then, of course, the question is, well, are there any identified untranslated features in the 5' end or in the 3' end of the mature mRNA sequences? So before I show you that, I do want to show you what this rat alpha tropomyosin, just for an example purpose, looks like in the NCBI database. So here, looking at the full record, this is the genomic graphic view here. And you can see by all of these lines that there are a lot of transcript variants. Some are predicted, as you can see, they start with the letter X, and some of them are known. They've been experimentally determined. So I would focus on, if you have the option for your gene, I would focus on the ones that begin with the letter N for known experimentally. Now, I've selected just one of these, and I've gone to the mRNA sequence for it. And that's this one here. This is just one of the variants. So this variant is called TPM 1.2. And what you can see here, as I have pointed out in previous videos, is that the gene sequence is shown here. This is the mRNA transcript that's shown in the top here. So that's the total length of this variant. This case, it's about 1,250 nucleotides. And then what you're looking at here is the protein. It codes a protein of 284 amino acids. Now, what you probably notice is that this protein sequence does not actually span the entire length of the mRNA sequence. So what I want you to do is to have a look carefully at the specifics. Where is that start spot and where is that ending spot? So to do that, up in this ruler section here, you're actually going to click and drag and zoom in on one region of your transcript. So when we look at this zoomed in, what's really cool is we can see right here that start site ATG that codes for methionine. And we can see which strand it's on as well. It's showing it on the upper strand, so that's going to be our template strand. And so that there is the start position for translation. So what I want you to do is to record how long is this 5' untranslated region for your transcript. So in this case, it looks like it starts at position 107. And that looks about right on the ruler here. So the first 106 nucleotides of this transcript are untranslated. Now, the other thing you can look at is whether or not there are any other features that are shown below. Some will have lots of features, and some genes, gene transcripts will have very few features. So for example, this one here has an upstream in-frame stop codon at position 83 to 85. And if you click on that, it will actually draw lines up to the sequence here, and you can look at the codon, the three nucleotide codon TAA. Now, you could always double check that um, in the conversion of codons to amino acids in that table and confirm indeed whether or not that is the stop codon that you're looking at. This is all within the untranslated region, so that stop codon is not going to have an impact on the translation of our protein sequence here. All right, so take note of anything in that 5' untranslated region in your gene. Then we're going to look at the 3' region that's untranslated. So if you use the zoom button right here, you zoom all the way back to 0%. Again, we can see the full graphic view. And now we're going to zoom in on the end. So from the end of the protein to the end of the mRNA sequence. And we can have a look here at now you'll notice because I didn't zoom in quite far enough, I can't actually see the nucleotide sequence. So if that's the case, zoom in a little bit further. And for now, I'm not going to worry about the rest of the 3' untranslated region. I'm just going to focus on the end of the protein sequence. Oopsies. So I'm going to zoom back out. Let me start again here. So I want to zoom into this region right here at the end. There we go, the tail end of the protein sequence right there. So have a look. TAA. There's that stop codon again. And indeed, that is where translation stops. So what I want you to do is to record what position that is at. And it will show you right here, location is 107. That matches what we were looking at the other end of the transcript. And it goes to position 961. And yes, right there, that's correct, 961. So take note of that position right there. Now let's zoom back out again and have a look at the 3' untranslated region. So there's a whole region from 961 nucleotides to the end that is not translated into a protein. That looks like the majority of this last exon here. And if you look carefully in this region, look for other features. And I like this one because it actually shows us a poly A signal. There is one in this transcript. And it also shows the start site for the poly A tail. So let's zoom into that region. We can easily calculate the entire length of this 3' untranslated region, but let's zoom in just to these features here that we're interested in so we can see the nucleotide sequences. So I'm zooming in again, and here we are. There's that poly A signal. If you click on it, it draws your lines up to the nucleotide sequences here, and you can see, in fact, that is textbook poly A signal sequence right there. So you can record that and record the position that is located in. And you can see that signal is just upstream of where the poly A site feature begins. So here, there's the A, and then there's your poly A tail that's showing for just the rest of that strand. So how long is your poly A tail? You can record that as well. So zooming back out, for this one, we've determined the start site of the protein. On the transcript, we've determined the stop site as well. And then you also calculated the length of the 5' untranslated region, also the length of the 3' untranslated region. And we also looked at this upstream feature within the untranslated region. And we also looked at these ones here as well, the poly A signal and the start of the poly A tail. So this is the example that comes from your textbook. And this is just one of the variants. You could go back, for those of you that do have multiple variants, you can go back to this record, choose another variant, and find out some more details about it. I'd like to also show you the example for the GRM6, which I've been using as my example. Now, this one looks as expected. It looks different than the other one. And what you're going to find yourself is that each one will look a little bit different. Some will have these details and these features that you can identify, and some won't, but all of that is information we can record. So for the GRM6, the full transcript length is about 6,000 nucleotides. And all the exons that are shown here includes information in the 5' untranslated region and in the 3' untranslated region. So what I want you to do is the same thing that you did that I demoed for the previous example. You're going to zoom in here and find the start site for the protein. So zoom in enough that you can actually see the nucleotides here. So ATG, there it is right there. And that looks like it's starting at position 62. And if you hold your mouse over the protein sequence here, it actually shows you exactly that. It starts at position 62 right there. So that means that the first 61 nucleotides are part of the 5' untranslated region. And in this example, there are some additional information or additional features that are included that weren't applicable to the other example that I was showing with tropomyosin. So for this one, there is a note here for the 5' untranslated region. And this is coming from the single nucleotide polymorphism database. And so that is a collection of sequences that have been in this, for humans, for example, which this comes from, is variations in sequence. So a single nucleotide difference between different individuals that have been identified. So if I mouse over on this example, it looks like at the position shown here that the nucleotide alleles could be either a G, a C, or a T. And if you go to the single nucleotide polymorphism summary, it probably will tell you just that. These features, of course, you could dive into all of the details of these features and learn more about them. You may also come across these in some of the literature that you might start reading in preparation for your discussion assignment. But certainly you don't need to go through and click on every single one of these and learn more about them. I don't see any other features within the first region. Oh, so okay, so unfortunately I wasn't zoomed into position 1. So we didn't see this first little bit here, which is good because that's thought we had lost some of our noted regions in the untranslated area. So there are multiple, these are the same. This is all from the same database. I was just telling you about that single nucleotide polymorphism database in the 5' untranslated region. So these are all the examples that are known. There is also a similar feature, an upstream in-frame stop codon, again within the untranslated region. If you click on it there, you can confirm TGA is indeed a stop codon. So let's zoom back out and have a look at the entire sequence again and zoom in into this region of your protein, find out exactly at what position it ends, and also determine the full length of the 3' untranslated region. So just scanning through this portion, and I'll just zoom in to make it much easier. So I'm looking at just the 3' region. There's the end of the protein right there. You'll notice that there is this region here, also from the database of single nucleotide polymorphisms. And these ones are less specific. If you click on them, it says variation heat map. And likely this untranslated region has is quite variable. So many different sequences have been collected, and they find that there are a lot of sequence differences in this 3' UTR. So you can just note those details. There are no specific links that you can go to and learn more about it if you find a variation heat map in your example. And then as far as any other features, there are none in the GRN6 in this 3' untranslated region. All right, so I think that covers all of the details for the mRNA transcript. And next week we'll start to look at the protein sequence, and we'll also start to look at some of the features of the protein sequence as well.